Slideshow to appear. Okay, so I think you should be able to see the presentation now.
Yes, we can.
Okay, fantastic. So thank you very much for that introduction. I can't claim to be an expert in COVID-19, but certainly I have some expertise in drug design. And we've been interested in this area because I'm a special investigator for the World Health Organization on malaria, and I get alerts from them every day. So as soon as the infection started in Wuhan, I had an alert, and we immediately decided to repurpose our work. So we entered quite early on, and we began to investigate to see what was available. So as I said, a particular interest is antimalarials, and the work that I'm going to show you is actually being done by undergraduates who change their work from synthetic work to computational work. So I'm going to do a quick whistle-stop tour of the main targets and why we chose particular targets that we thought we could do something with. So undergraduates were involved in this work. They wanted to do some work for combating the virus, and they wanted to repurpose our antimalarials, which we already had. We've got our in-house private library of around about 100,000 compounds. We're also interested in identifying existing natural products and decide which, if any of these, could serve as a platform to design new drugs. We decided to use freely available programs such as Autodoc, Vena from Scripps, and local computational resources, because the ones that we normally use, for example, those in Switzerland, were in fact completely unavailable because of large numbers of queues from people all around the world. So you may think of this as a David versus Goliath approach that we were taking. So much of the work has already been described in this conference by people who are much more versed in this area than I am, but nevertheless, I'll still go through very quickly the genome to summarize what's already been said. So it's around about 30,000 base pairs in length. It codes for 2 large polyproteins, PP1A and 1AB. There are 4 structural and 16 non-structural proteins. And of the structural proteins, there's a spike protein, which we've heard much about, the membrane protein, an envelope protein, and a nucleocapsid protein. So we had to decide which, if any of these, would be attractive targets. And most people have gone for the spike protein when they're working with immunology. So if we look at potential candidates for medicinal chemistry, there's 3CL-Pro. It's a chymotrypsin-like protease, and it's releasing non-structural proteins. There's also PL-Pro. It's a papain-like protease that cleaves polyprotein at 3 sites. So this is of considerable interest to us. There are also others which we weren't too familiar with, the RNA-dependent RNA polymerases, the S protein, and ACE2, which you've also heard something about. And of course, if ACE2 is not expressed, it's felt that COVID is not so much of a problem, and this is the case in younger individuals. And there's also TMPRSS2 that we heard about earlier on today. So that's important for cell entry. Because in terms of drug design, we wanted to be able to design against known enzymes, we were particularly interested in crystal structures that have been deposited. So these are either single crystal X-ray diffraction or those from cryo-electron microscopy. So if we look at this particular PL-Pro, it's 83% conserved from the previous infections with SARS-CoV-2. And this is why there was success of the vaccine, because a lot of work had already been done. And the media hasn't actually emphasized that, so there's some vaccine hesitancy. The active site consists of a catalytic triad, cysteine 112, so it's nucleophilic. We've also got a basic histidine 273 and an aspartate 287. So the mechanism of action is the same as 3CL-Pro, and we'll have a look at the actual structure graphically later on. And this is important for alignment of various histidine residues. And the enzyme also breaks down ubiquitin and 1SG15, which triggers the host immune response. which we've heard about in terms of the COVID storm. So in SARS-CoV, this enzyme prefers ubiquitin, but in SARS-CoV-2, it prefers a different substrate. So we were particularly interested in deciding which one of these we were going to decide to target. So looking at the next protein, we were interested in particular areas of this particular target. And those of you interested can actually download some of these structures. Inhibitors have been made which block this particular active site, and we could target RDRP inhibitors, as many nucleoside analogs exist, and of course some of these have been repurposed. So an example would be favipiravir, which is already approved for use in Japan. So the structure that's shown up there, and I'm sure you will recognize its analogy to genetic structures that exist within us. The next protein that we were particularly interested in was also deposited in the PDB. So this is a protein target. Biologics mainly target this particular ectodomain. And we thought this would be a little bit difficult, especially for the small computational power that we had. And other groups were looking at this, and it's mainly immunological work that's been done in this area. And finally, just to say a little bit about ACE converting enzyme 2, we've done a lot of work in this previously in a different concept, different context, and we already had a number of known inhibitors, but we felt that working in this area, which was initially attractive, for example, chloroquine is supposed to act at this particular site, we felt this has been discredited, especially by a certain president who also wanted to expose you to large amounts of light, but also wanted to inject you with bleach. So there may have been some merit in this particular area, but the doses that we felt that were being used, despite it being an anti-malarial, weren't borne out in clinical trials. So there are also problems if we block this site, because it induces hypertension and glucose intolerance, which did cause damage in quite a number of patients until it was realized that this was happening. So we felt that even though this was an attractive target and we had antimalarials, we really wanted to move away from this area. There's also transmembrane protease serine 2 that's been previously talked about. And this allows accumulation of basic compounds, and this may also be a target for chloroquine. And we thought this could be quite important because our group mainly works with alkaloids. We felt that this could be an area that we could possibly do some work on. This also primes the S protein, and the biochemistry of this is extremely well established, which you can look up if you're particularly interested. Cathepsin L isn't highly expressed in Calu lung cells or throat epithelial tissue, so inhibition of this could still block entry into the cells, preventing infection, but we felt there was insufficient information for us to be able to target this. So we became interested in the main protease. So just a little bit about the actual enzyme. There's 93% conservation from SARS-CoV-2, so we thought this was something that we could look at, especially computationally. It exists as a homodimer. Most of the modeling is actually done with 1/2 of the homodimer, and there's a catalytic diet consisting of cysteine 145, which actually makes a tetrahedral intermediate here. So we thought that this particular site was attractive, as have other groups who've done extensive molecular modeling, especially using multi-scale methods, which require extreme computational resources. Certainly, we didn't have access to those. So if we look at the SARS-CoV-2 active site and the SARS-CoV active site, I hope you can see that there are similarities, but we can see there also visually, you can see there's some changes here. So you can think of this as a sort of a U-shaped cleft here, where there's less of a U-shape, if you like, here. And these have been numbered for convenience: S1, S2, S3, S4, and S5. And we became particularly interested in the site because we thought it was druggable. So consequently, our group thought of using autodock RINA, as we indicated previously, because this gives us a docking score. And the students like to sort of think of how comfortable this ligand would be in the active site, because we do outreach work as well, and we wanted to be able to explain this to the general public. The type of output information that you get is a Gibbs free energy function. We get it in kilocalories per mole. And the more negative the docking score, the more comfortable the ligand is. But it's important to remember, just because it's comfortable doesn't mean to say it necessarily inhibits that enzyme. It just means it's a good fit. We then found that a study had appeared in Brazil, which identified one of our main antimalarials, which is in fact much, much more potent than chloroquine, known as metaquin. And they had found that, in fact, this was a very good target for 3CL-Pro. So this particular piqued our interest, and we, in fact, started a collaboration with these people in Brazil, which I'll be talking about later on. And we thought, well, there might be some opportunities for non-covalent inhibition of these particular sites. However, I must emphasize that we do know that this particular compound in malarial parasites acts by a different mechanism. There's actually electron transfer occurred, which may not happen within a coronavirus. We didn't actually have biological testing facilities available, even though we approached national centers. It wasn't available, and I'm sure many other people were trying to do the same thing. So we looked at UCSF Chimera, and those of you who are interested, there's a lot of YouTube video material available, so you can start working in this area. If you're particularly interested, you can download autodock RINA. But it's important to prepare your actual protein structures properly, remove waters, make sure that the sites of protonation are correct, etc. I won't go into the details, but if anybody's particularly interested, you can have a look on YouTube. So we prepared all our molecules using ChemDraw and Chem3D, which is freely available to our undergraduates, and they could access this from their home computers. And then this was imported into Chimera, and we began our studies. So we benchmarked our work using peptomimetic inhibitors, which had already been made. And we looked at this, and in fact, the Zhang group has provided information about close contacts in this target site, and you can read about it. This is published in Science. So this is really a landmark study that had occurred. So if we quickly look at the synthetic designs that are available, when designing drugs to fit into these protein targets, the actual cost of taking the drug to clinic can be around about 2.6 billion dollars, and that would include full ADMET and clinical trials, toxicology, etc. And you also need a component for litigation in case the drug causes damage, and that's considered quite important. So many people have moved towards repurposing, but the actual chances, if you look at the statistics, is only 10% to be able to repurpose something. And this is because drugs these days have been decided not to have as many off-target effects. So that's not unsurprising. So we previously had some information in one of the talks about the TMPRSS2 inhibitors. I won't talk too much about it. It was known as FOI305 previously in Japan. It's been known since the 1980s, and it actually treats inflammation. And it's one of their favorite candidates for drug development. But it doesn't fully block cell entry. So we wanted to move away from that particular area. And as I said, there's some evidence that chloroquine could act there, but the concentrations needed really are moving into toxic doses. A lot of important work has been done by the Ghosh group, who are experts in SARS-CoV. And they've published an excellent review, which I suggest that you look at. And a variety of inhibitors were available, and these are in fact covalent inhibitors. So if you look at the actual values, they may appear not to be so effective, but because these are covalent inhibitors, you can't really compare the values with non-covalent inhibitors, which we're interested in. And the enzyme, as I mentioned, we've got that thiol group, which interacts with a carbonyl shown at the left-hand side of the page here, forming this tetrahedral intermediate and releasing a small molecule. So a number of attractive compounds have appeared. Well, the one they particularly favor, in fact, is this one, which is their hit compound, which they've actually looked at analogs of, and this was published in the Proceedings of the National Academy of Sciences, the GRL 0617, but a lot more work needs to be done. And if you actually look at the shapes of these, you can see that they have that sort of U shape that I mentioned. And they need further development toxicity testing. Of course, Ghosh is one of the people who's been an expert and has a drug against HIV. So I'm sure they'll come up with something pretty soon. They've been fairly quiet at the moment. So because of all this previous work that had been done for HIV studies, the main attraction for synthetic compounds have been peptomimetic type compounds. And we previously had some information from Touraj to show how compounds that inhibited the immune system could also be useful, but they're in fact cyclic, but they also have high amounts of toxicity. These particular compounds aren't, and they have attracted the attention of Pfizer, and many of these are covalent inhibitors, especially if you've got this this particular type of grouping here, which is particularly popular because it actually cleaves off and chemically labels the inside of the active site. So one of the main successes of repurposing drugs has been remdesivir, although there are in fact prodrugs which may in fact be better, and some of this work was published in the American Chemical Society talks earlier this year, which I attended, and I encourage you to look at those if you're interested in this particular area. This comes from Gilead Sciences, and they've made a considerable amount of money. And people you may have heard of who've been infected, unfortunately, in India at the moment, are trying to source remdesivir, but it's in short supply. So in a cell-free environment, the EC50 is around about 0.77 micromolar. And if you want a sort of a benchmark of the sort of values that you really need for good effectiveness, you really need it in the nanomolar region for the compound to be selective and active. So if you now move on to natural products, we looked at all sorts of various oils. We also were cognizant that to develop a new drug could take a considerable amount of time. But of course, we could move forward fairly quickly. So we began to look at a variety of databases, and students were particularly interested in direct delivery to the lung. And we've actually done some work in this area. So we looked at a variety of compounds such as ashwagandha, giloy, and holy basil. And so we looked at the chemical components of these. So we had some work earlier that showed that with analytes, we had actually looked at these compounds before any work had been published. So the work of Tripathi was published much later, and their values for targeting these docking scores, in fact, were much higher than the ones that we've got. And this is one of the limitations of using different types of modeling. So we were aware of that, and we really wanted to think how we could compare our work with that of others as well. So if we look at a variety of other compounds, one of the compounds that we were about to have a talk on, but it got cancelled, was hesperitine. So there's a possibility of using that. But the other ones that we found that hasn't received much attention is St. Johnswort, and we felt that this particular compound could be looked at. And we looked at a variety of substituents, and we had a reasonable docking score, but we also felt that there was some toxicity effects. So we moved on to these. But this targeted enzyme that we weren't particularly interested in, the NSP8 and NSP12 linkage blocker. So there are other compounds, and you'll note that these are also U-shaped compounds. So I'd encourage other people to look at these particular compounds because they fit into the active site better. So we have a particular hypothesis as to why these would work. And in fact, they've got quite good docking scores. So I would suggest that the compound that we really should be looking at, apart from mentoflavone, are these two, getting, and of course they're found in various natural products, plants, and are of considerable interest. So we made sure that we looked at all docking studies that we could get our hands on, and we cross-correlated our results with
2 minutes, Fyaz.
And so we found that, in fact, there was a linear plot, but our particular compound that I mentioned, metaquin, was in fact superior. So we decided that by benchmarking known compounds and training our AI program with these particular compounds, it was possible. Quite an important piece of work appeared from the Jorgensen lab, and I would encourage you to look at this. And this is one of the first compounds, a modification of a compound, a good medicinal chemistry study where the main protease is being attacked by parampanel. Parampanel does have some adverse effects, but they've published some of this work in ACS Central Science, which I suggest that you look at. But the advantage they had was they had an iterative bioassay-based model with live virus, which we didn't have access to. So we could only look at our compounds computationally. So together with our colleagues in Brazil, we took our compound metaquin as a starting compound. And after around about 10 AI design cycles, we have a compound with a sort of binding efficiency of 14 kilocals per mole. It binds at 14 different hydrogen bonding sites. And we believe once we actually synthesize this compound, it may have good activity. One of the other compounds which has been highlighted, in fact, is berberine as a nanoparticle solution, but there are off-target effects with berberine. So coming up to the conclusion, we found that natural products can provide an immediate alternative design. We've got chromophore, but these are rarely good inhibitors in themselves. And of these, as I've indicated, a particular compound shown on the left, berberine. But whilst our study was in progress, Pfizer's new oral candidate came around, and work has been published on this, and it's in clinical trial. And this may be the new oral candidate that may be useful for combating the current strains, but it may be ineffective against drug-resistant strains. So we think natural products could be important in terms of repurposing, but we've yet to find a compound with that sort of nanomolar activity that we need. I'd like to thank the students who did the work, Ebony Dickinson and Olga Singo, and it was Joe Moylan who actually wrote most of this talk, because he likes these sort of colorful slides that move around. And we also had Alexandra Kelly Hunt, who did a lot of the work, and my PhD student Said Alizadeh, who's about to finish. I'd also like to thank Professor Ricardo Ramos, who indicated our compound metaquin was very active. And we in fact started a collaboration with him, and we'll be looking at the compounds at Fiocruz in Brazil. So we've in fact got all of the proteins modeled there, and we're doing in fact a very, very large study where we're screening thousands of compounds against every receptor and looking at compounds. So finally, I'd like to thank you for your attention. Sorry about going through things so quickly, but in the short time available, I think that's all that could be done. So I'll be happy to take any questions. Yeah.